All right. Well, good afternoon, everyone. Thank you for joining us at the MAPP Rounds. And today I have the really great pleasure of introducing Dr. Keenan Walker, who is our speaker today. So Dr. Walker is a tenure-track investigator at the National Institute on Aging Intramural Research Program, and he is also the chief of the Multimodal Imaging of Neurodegenerative Disease Unit. He did his PhD in clinical psychology at Johns University with a focus on clinical neuropsychology. And then he completed his internship in clinical neuropsychology at the University of California, San Diego VA site. He then completed an NIH-funded postdoctoral fellowship in dementia and geriatric cognitive disorders at Johns Hopkins University School of Medicine, and transitioned to the role of assistant professor of neurology after receiving an NIA-funded K-23 Career Development Award. Shortly thereafter, he began as a tenure-track investigator at the NIA. Dr. Walker's current research program focuses on understanding the role of abnormal immune function in Alzheimer's disease and late life cognitive decline. And so before we get started, I just wanted to let you know, please write your questions down or type your questions in the Q&A, and Dr. Walker will address those questions towards the end. So Dr. Walker, thank you again so much for joining us. We're really excited to hear about your research, and you can get started.
All right. Thank you, Marie, for the introduction. I appreciate the invitation. It's really a pleasure and honor to speak with your group. So, yeah, I'm going to talk about plasma biomarkers, and probably in a different way than you may have heard, really focusing on this untargeted approach to biomarker discovery and how we can use that to learn about disease mechanisms and, predict disease itself. So I'm going to go ahead and start with this basic framework that that my lab has, and we're not unique in this, but we think that conditions outside the central nervous system, systemic conditions, can influence risk for Alzheimer's disease and dementia more broadly. And we know this from epidemiologic studies, that have been done for decades and decades, that things like diabetes, hypertension, heart disease, autoimmune inflammatory conditions can all increase risk for dementia and Alzheimer's specifically, in varying degrees, vascular dementia as well. Now, I include this figure on the right from the Lancet Commission in 2017, really just to illustrate that anywhere from 30 to 40% of the attributable risk for Alzheimer's disease is thought to be due to the conditions that lie largely outside the central nervous system. So really, these things are modifiable, and we think that proteins in blood might actually mediate this relationship between systemic disease, again, non-CNS disease, and dementia risk itself. And the framework that we put forth is provided on the top. Let me find that laser. So we go from clinical or subclinical disease, or just non-disease processes altogether, like cellular aging. We know these conditions can influence protein expression, not just in specific tissues, but these proteins then find their way into circulation. Now, these proteins can influence target cells in the brain itself through various conduits. We've heard of the neuroimmune axis, includes things like blood-brain barrier, choroid plexus, afferent vagus nerve signaling, etc. But a number of these proteins have already been identified. These are proteins in blood that circulate, that can influence what happens in the brain. A lot of these happen to be cytokines and chemokines, but we think there are many, many more out there, that is proteins in circulation that can be discovered that likely have some role in determining dementia risk. Now, we pursued this hypothesis, and it became more pursuable with the advent of large-scale proteomics done in a way that's scalable, cost-effective, and uses minimal amount of fluid, biofluid. And so you may have heard in recent years, platforms like Olink and SomaLogic have become more and more popular. And it's kind of like we're in the age of the proteome, where we were in early 2000s, for genetics. It's expanding rapidly. The price is coming down, and you're seeing more and more cohorts adapt this technology. But we adapted the SomaScan platform in the ERIC study back in 2016, about 2017. And from that, we did one of the earliest large-scale plasma proteomic studies of incident dementia risk. And let me just stop and so we're all on the same page. When I say proteomic platforms, I mean platforms that measure large swaths of the proteome. If we think of about maybe 20,000 protein coding genes exists, we're talking about platforms that measure a big chunk of that. So some of the earlier versions of SomaScan, this is our proteomic platform measuring more than 5,000 to 7,000 proteins. Now 11,000 proteins can be measured, and this is all with like 50 microliters of blood. So we applied this to our large cohort, and we found a number of proteins that predicted dementia risk in a 5-year follow-up period. A lot of these proteins were novel, and what was more exciting was we identified some proteins that we thought were mechanistically relevant, and I can tell you a little bit more about how we did that. But there were a number of studies that also came out around the same time that looked at late life dementia risk, measured proteins in older adults. And one of the limitations of our study and all these contemporary research reports was that Alzheimer's disease really has this protracted period, and we were looking at late life. Now we know that the pathogenesis of AD and the pathogenesis of non-AD dementia is thought to begin decades earlier, right? We have this preclinical period where we have amyloid, tau in the context of Alzheimer's disease that is accumulating without the presence of symptoms. And really, in order to understand the pathogenesis, it's thought that, well, we have to look as early as midlife when the process is actually getting started. This is for Alzheimer's disease, but not exclusive to Alzheimer's disease. So we think this happens with vascular dementia and some of the synucleinopathies also. So we were focused next on looking at this midlife period. And so what we did in a more recent paper that came out middle of last year was we looked at plasma proteome in middle-aged adults and related proteins to incident dementia risk, really to identify midlife drivers of dementia risk and identify early biomarkers. And so again, we started with the ERIC study, and this is the atherosclerosis risk in communities cohort. This is a multi-site community-based study done at 4 different sites across the US. And it was started in the late 80s, actually. But we took blood from visit 3, which was in 93 to 95. And at this point, there were about 11,000 individuals, who had blood available for measurement. In the ERIC cohort, it's pretty much 3/4, American white population and 1/4 African American. So it's diverse in the sense that it's multiracial, but keep in mind that this study back in the 80s was designed to be a population-based study of white and Black individuals in the US. So keep that in mind when interpreting all the data. But what we were doing here was we took 11,000 samples, measured our 5,000 proteins using the platform in these 11,000 samples, and one at a time related the abundance of each protein to dementia risk over this 25-year follow-up period. So in that time, in that follow-up period, we had about 1,900 incident dementia cases, which gave us a lot of power to detect effects. And so here is our first set of results from that analysis. And this volcano plot essentially summarizes what we found. We found 26 proteins that met this bond for any corrected threshold of GDF15 rising well above the rest, with 25 others. And this is with adjustment for covariates listed here: demographics, cardiovascular risk factors, kidney function, etc. So we really were interested in finding proteins that independently predicted dementia risk. And again, these proteins were measured in people in their 50s and 60s for the most part. So we looked, then we asked, okay, what proteins in their 5th and 6th decade predict near-term dementia risk? So more incipient dementia. And so we capped our follow-up time to 15 years and ran the same analysis. And here we found GDF15 come up again, but in addition, we found 5 additional proteins or 6 additional proteins that were associated with dementia risk in this short-term period that didn't meet the threshold for statistical significance, over the full 25-year follow-up. We did the same thing. Well, we did something similar, but looking at now at long-term dementia risk beyond 25 years to say, okay, what proteins are really relevant across this longer time horizon. And we found GDF15 come up again with 5 others. These 5 actually were identified in the primary discovery analysis, but we can say that these 5 in particular seem to be early movers in people who are at risk for dementia, as much as 2 decades later. So when we put everything together, we find 32 dementia-associated proteins that we can pick up in middle-aged adults. And they're represented here with GDF15 being significant at each of the follow-up periods. So 4 of our proteins have already been nominated in the Accelerated Medicine Partnership, which really takes a totally, largely different approach to target identification. So we're happy to see this overlap in these potential novel targets. So I mentioned, maybe I did mention the mean age of our population when the proteins were measured was 60. So, we're teetering on sort of the later side of midlife. What we wanted to do next was answer the question, do we have bona fide midlife biomarkers? So we luckily had a large enough sample that we were able to look at the younger half. So basically people in the 4th and 5th decade of life with the same set of 32 proteins across the same follow-up periods. And you can see the results from our primary analysis in blue versus the the younger midlife analysis in red. And you can see a lot of the associations are preserved and some are even amplified. So we do think we have bona fide midlife dementia biomarkers here. That may be detectable as young as, people as young as 40 and 50. What about the biology of these proteins? So, qualitatively, we can say they kind of fall into one of 6 different categories, with immune function and proteostasis and synaptic function probably being the most well representative. We did the additional pathway analysis. I'll talk about some network analysis that we did. So really important for these biomarker analysis is validation, especially when it's biomarker discovery. And so although there's not a whole lot of cohorts out there like the atherosclerosis risk and community study, we were able to approximately validate some of these biomarkers using different study designs, but studies that had the same proteomic platform, importantly. So we looked at the EMIF cohort, we looked at the Whitehall 2 study. EMIF was cross-sectional. We found a large proportion of the proteins that we identified at midlife as being relevant to dementia risk were differentially expressed when we compared to control to AD, and when we examined which proteins among MCI people predicted progression to AD. And then in Whitehall 2, we were less powered here, but we found about 5 of our 32 proteins predicted 20-year cognitive decline. Now, one of the things we could do with EMIF, this cohort, was look at how our proteins were associated with some of the neurobiologic changes that we could proxy through CSF biomarker measurement, because this cohort had the same panel of proteins measured and concurrently measurements of amyloid tau and some of the other dementia or neurodegenerative biomarkers. So here is our list of proteins measured in plasma again, and CNS biomarkers again measured in CSF, and everything that's in gray just wasn't measured in the cohort or didn't pass quality control. But we see for the proteins that were measured, a good proportion of them do show a positive or negative association with amyloid status, p-tau levels, and total tau levels. And we were able to look at CSF NFL, and neurogranin as non-specific markers of neurodegeneration, and found some broadening of the effect here to a few other proteins that what we think are neurodegenerative related, but maybe not Alzheimer's disease related. And then YKL-40, which is our, I think, astrocyte-mediated marker of neuroinflammation. GDF15, interestingly, I'll just point this out as an example, was the strongest association with dementia risk, across all 3 follow-up periods. It was validated in 2 external cohorts as being clinically relevant, but we're seeing no association with A beta, total tau, p-tau, but we do see, again, positive association with our marker of neuroinflammation. So, we're getting a sense of what these proteins, if they are indeed mechanistically relevant, what pathways they may be operating through or not operating through. But GDF15, our strongest candidate, seems to be operating through immune-specific processes. So where do these proteins come from? And we could answer that question to some degree using publicly available data sets like GTEx and Human Protein Atlas. And so, we ran all our proteins through these databases and got these very busy heat maps, but there's really 3 points that I want to make. So proteins can either, we found a big subset of proteins that were non-specific. So they were produced by most, if not all, tissue types at the gene and protein level. We found a subset of proteins that were CNS specific. So these were produced primarily or exclusively by brain tissue. And so these are things like complexin 2, complexin 1, or wherever it is on the heat map. But again, and it turns out, as we might expect, a lot of these are synaptic proteins. And then we see some proteins that don't seem to be expressed, at least at measurable levels, in brain tissue at all, yet have strong associations with dementia risk and presumably other CNS outcomes, including GDF15. So we're not seeing it expressed in brain. We know it's expressed in brain to some degree from other research, but here we think it's mostly operating peripherally. Now, for those proteins that are expressed in brain, we could go to other data sets that have done brain tissue proteomics and see if they're differentially expressed in autopsied AD brains versus controls. And we did see some evidence for this for those that were measurable in tissue and brain tissue. And really, the upshot here is the stress response and immune proteins upregulated in AD brains, and our synaptic proteins, complexin 2, complexin 1, and cerebellum 4, are strongly downregulated. And interestingly, this is the opposite direction, at least for our synaptic proteins that we saw in plasma at midlife. So we might see this depletion of synaptic proteins in brain tissue, at least at the terminal stage of disease. And in our 40 and 50-year-olds, these synaptic proteins seem to increase their levels in blood well ahead of dementia onset. So we can understand more about the biology with our network analysis. And we did this with the help of Pascal Schlosser and Linda Zhao. They were at Hopkins at the time. And so we used a platform or a package that was very similar to WGCNA that some of you may be familiar with. This is called NetBoost, and it's really just a data-driven clustering platform that allows us to, based on gene or protein expression, identify modules of co-expressed proteins or genes. And so we applied this to our full set of 5,000 proteins that we measured in the ERIC cohort, and came away with 19 modules that are represented here in this dendrogram. So they range in size from anywhere from 20 proteins to 2,000. So a lot of variation in module size. But what's neat about these modules is that we can characterize the person-specific module expression and then relate module expression to various outcomes. And of course, as you might have guessed, we've related module expression back to dementia risk. So again, these are protein modules or groups of proteins, and we think are biologically relevant that are expressed in midlife, and we examined their association with future dementia risk over 25 years. Now, module here, these are arbitrarily named. Module 1, 5, and 19 were associated with this 25-year dementia risk. Module 9 associated with near-term dementia risk within that 15-year period, and then 1, 5, and 19 again, long-term dementia risk. So we found these 4 different modules with after covariate adjustment and FDR correction seem to have this robust association with future dementia risk. So we looked further into what these modules represent. And essentially, these are groups, again, of co-expressed proteins, but we can run them through annotation platforms and try to get a sense of what biologically they may represent. So this 700 protein module, module 1, which was downregulated in context of dementia risk, seems to be largely enriched for immune processes. JAK-STAT signaling, TLR, ligation. You see a number of, probably familiar immune processes here when we look at our annotation. Module 5, 200 proteins enriched for extracellular matrix organization and degradation. 19, weaker enrichment. It's a smaller module, 77 proteins, but we see some modest enrichment for immune processes. But we can look at our hub proteins, which are the most module specific. They're most highly correlated with the module itself. So yeah, they're essentially hubs. And we see a lot of heat shock proteins here, suggesting stress response. And that's what we think this module may represent, and hence the immune pathways that are annotated here. But notably, module 19 is shows the strongest association with future dementia risk. And then for module 9, which is associated with dementia risk in the near term, we see something different. It's coagulation and complement signaling. And the directionality is a little tricky because we have downregulation and then downregulation, versus upregulation. So I tried to do the mental gymnastics for us, and also put these on a time, pseudo time sort of, but not in a formal sense. So we have this multi-decade evolving immunologic signature that we could sort of pull out of this. But what we're seeing from the module enrichment is that early, those modules associated with dementia risk over the full follow-up period and the longer-term follow-up period, we see this involvement in JAK-STAT signaling, leukocyte activation, toll-like receptor signaling. Not necessarily all activated. We see this downregulation of antiviral signaling and and adaptive immune differentiation, which is really interesting, and it allows us to get at the complexity of how early immune function, how early inflammation may influence dementia risk. And it's clearly not so simple with sort of immune processes operating in opposing directions. And then we see module 9, which was associated with more near-term dementia risk, really enriched for complement coagulation as well. And so that seems to be perturbed as we get closer and closer to symptomatic presentation. So then what we did was we wanted to understand, okay, are these proteins or modules causally linked to dementia risk? We know we have associative relationships that are pretty strong. So what we did was we worked with Mariam Fornage and Yunju Yang at UT Health Science Center. They were able to run a genome-wide association study on plasma protein level and protein module expression. So we identified genetic variants that were associated with the level of protein abundance in plasma and the level of protein network expression in plasma. And then we were able to use these, what we're calling protein quantitative trait loci, and see if they were relevant, as predictors of Alzheimer's disease. And this is the Mendelian randomization, naming 2-sample Mendelian randomization framework. We also looked in the opposite direction. So I'll breeze through these. The summary of what we saw when we examined if proteins were causally linked to Alzheimer's disease using this Mendelian randomization framework was that we saw evidence for 2 proteins, calcitonin 3 and serpinate 3, as at least putative causally linked to AD from this, using this approach. And I'll talk a little bit more about this because we're going to reference it later. But essentially, it relies on this random allocation of genetic variation throughout the population. Some people have variants that code for high levels of serpinate 3. Some have genetic variants that code for low levels. And so you're exposed to higher low levels of serpinate 3 throughout your lifetime based on this random distribution of alleles. And so in a sense, it's like nature's clinical trial. And because of the timing, we don't have issues of reverse causation. Because of the randomization, we don't have issues of residual confounding. So, with some assumptions, we can make causal inferences using observational data in this way. So it's a really powerful technique, led to the discovery of multiple drugs. And right now it's implicating serpinate 3 especially, and this is with all the sensitivity analysis as causal. We looked in the opposite direction also. Is Alzheimer's disease associated with protein level? And we found a lot more evidence for that. So it seems that the disease itself is influencing differential expression of proteins in plasma. But it's like, okay, well, that's not that, obviously a big change in your health status, proteome changes. But keep in mind that this is 4th and 5th decade of life. So we're seeing the Alzheimer's disease biology, or perhaps Alzheimer's disease genetics, play out, in in middle age, at the protein level. And here we, in addition to finding that, we genetically validate the role of some of these proteins as biomarkers. Maybe not specific to Alzheimer's disease, but it does support their sensitivity. So all in all, we have this set of biomarkers that we think represents different aspects of biology that, we think are relevant in midlife and are predictive of Alzheimer's disease. And in some cases, well, let me start over, predictive of dementia risk, and in some cases, Alzheimer's disease. Now we have proteostasis, immune function, synaptic function, and ECM organization. They seem to be the most, common. So we think to the degree that Alzheimer's disease is a heterogeneous process, and biologically speaking, and there's more and more evidence to suggest that that's the case, we think some people will have higher, be more, show more synaptic dysfunction versus more immune dysfunction. And we think these proteins can give a sense, okay, what's your dementia risk, and on what sort of etiologic pathway might you be on. So we examine this more formally, like how well do these proteins actually predict dementia risk, in a paper that's under review right now. But we essentially tried to identify the optimal combination of plasma proteins in blood that in middle-aged adults that predict dementia risk. And they were named soma signal tests, so I'm going to refer to them as such, or SSTs. And that's because we partnered with SomaLogic to develop this biomarker combination, and they gave it its name. But this was a project that was led by Michael Duggan, who's a postdoc in my group, and Claire Patterson, who's a scientist at SomaLogic. But essentially, we split the ARID cohort into a 70-30 training and test. It was really 70 training, 15 tuning, and then 15% validation. So because it was large enough, we were able to split it up. And then we developed the model in the 70%, and then tuned it, and then tested it in a 15% holdout sample. So I'm going to breeze through the results in terms of how well this dementia soma signal test, or dementia risk score, did for dementia prediction. But here is the area under the curve, and I think, that's a good benchmark, especially when you're identifying stuff that exists already, when you're diagnosing something that's currently present. But I think the bar is a lot higher. Well, it's a lot more challenging to identify dementia 20 years out. So, we think there's some value in these, okay, not great, not stellar AUCs or C statistics. And maybe I think the next slide illustrates it somewhat better. If we break up the midlife dementia risk score into 4 different bins, and I forgot to say, this dementia risk score is made up of 25 proteins that were selected, using a machine learning approach. I just didn't have time to go into the actual selection, but think of this as a 25 protein score. And these aren't necessarily all the same proteins that were highlighted in the last paper. Some are further down the list that happen to contribute some unique variation to prediction. But so we can bin people into 4 different bins based on their midlife dementia risk score, from low to high. And you can see, difference between someone in a low bin and someone in a high bin is about 10-fold. So if you're in a high bin, we think you're 10 times more likely to develop dementia over this 20-year period than in the low bin. And the observed versus expected, was fairly consistent. And so, we have this risk ratio of 10, which, so we have less ambiguity there, with more ambiguity if you're in the median groups, which, happens, right? We tried to examine, okay, how well does this do against something that we can consider a gold standard, or something that people are more familiar with in terms of biomarkers. So although this A beta and p-tau, these biomarkers weren't actually designed to predict clinical progression, more so like pathology, we examined how our dementia risk score compared to those, along with NFL and GFAP and predicting 20-year dementia risk. And you see the results here. So this is in a subset of the sample. So we're still coming in at 0.7 for our dementia risk score. But if we look at midlife A beta in plasma, midlife p-tau 181 in plasma, NFL and GFAP also in plasma, by comparison, we're doing better with this dementia risk score. If we combine everything, we can get up back to low 0.7s. So we are outperforming some of these gold standard metrics. We don't have p-tau 217, unfortunately. We are applying it to the cohort, and I think that'll be the next comparator, because I know there's a lot of justified hype around the value of that biomarker. But we looked at the same risk score applied to older adults. So we're moving from 50 to 60-year-olds to 70 and 80-year-olds, and it actually does a lot better. So it was recalibrated to some degree, but same protein, same beta weights, and we're approaching 0.8. So it's much better predicting 5-year dementia risk among older adults. And it maintains a strong prediction about 0.77 in validation. The risk ratio from low to high is, it was 10 at midlife, it's now 13.5. So the score performs actually better in older adults than it does in middle age. But, shorter follow-up period, more dementia cases, that's a more favorable, setting for developing an accurate risk score. So how does it perform in relation to A beta, GFAP, p-tau, and NFL at late life? Well, it still outperforms, and I was actually surprised by this, it outperforms these measured among older adults as well as middle-aged adults. And when you combine it, we can reach about 0.8 for 5-year dementia prediction. So, that we hope to be published soon, but that's our long-term dementia risk score. And, I don't know personally if that, I necessarily want to know my dementia risk at age 40, 50, or 60, especially with what's out there currently in terms of interventions. But, I have heard from the company that's marketing this, and it's marketed as research use only, but it's been clinic there. I don't know. But we think we can optimize the score, and we're working on doing that. We want to achieve better prediction, and we think we will if we can, really identify etiology-specific dementia. I think one of the limitations was we were looking at a heterogeneous group of all-cause dementia, and of course, we think there's different proteins associated with AD versus VAD versus LBD, for example. So that's that's in production right now. Now, I'm going to switch gears and talk about more of how we try to identify more so therapeutic targets or potentially causal proteins. And going back to the, implementing our genetic approach to do this. So here we are working on identifying genetic loci that increase risk or dementia risk through their effects on the plasma proteome. And so this, the framework here is that, again, genetic variation influences protein expression, and then proteins in circulation can then influence what happens in the brain. So this was done in collaboration, again, with Pascal Schlosser and a few other individuals, namely taking the baton from Zhang et al. And Nilanjan Chatterjee's group at Johns Hopkins. What they did was they used the SomaScan proteomic platform in ERIC to identify a number of protein quantitative trait loci. Again, genetic variants that predict protein level, but they did this for all 5,000 proteins that were measured. And they looked specifically at genetic variants within the region of the cognate gene. So that's why we're talking about cis pQTLs, and rather than trans, that are variants that are outside the protein coding gene region. So they looked specifically at these cis pQTLs, identified about 2,000 of them. Well, identified cis pQTLs for 2,000 proteins in our European American cohort in ERIC, and then for 1,600 proteins in our African American. So this was, at the time, one of the largest pQTL discovery efforts and I think still is for African Americans. So this gives us a very unique and powerful tool because now we have these genetic proxies for protein level that we can then apply to look causally at how proteins might, really predict or be mechanistically relevant to specific diseases. So we took these cis-pQTLs that were, identified in this unique way. I encourage, if you're interested, this is a great paper. But they really took this combination of cis-pQTLs, applied machine learning, and found the combination that best predicted protein level. We then took these genetically proxied protein levels and applied them to Alzheimer's disease GWASs. We looked and we took the European American pQTLs, applied them to the Bilinguage GWAS, which came out in 2022, and then took the African American pQTLs and applied it to the Kunkel GWAS, which was an African American Alzheimer's disease GWAS that came out in 2020. So here was the results from the European American pQTLs applied to the Bilinguage GWAS, and we have magnitude more power here. So I'm going to focus more so on these because this is where we found a lot of our results. But as you can see, when we did this, and again, this is genetically proxied plasma protein level against, genetically proxied Alzheimer's disease, essentially, but it's in a way that allows us to make this causal inference for the reasons I mentioned earlier when I was talking about two-sample Mendelian randomization. So we have 18 proteins that came up as significant at this Von-Brony corrected threshold. And we found 6 of them, those bolded here, hadn't been identified in GWAS studies previously for Alzheimer's disease. So we were excited about this. And of course, we're taking a unique approach to do this. And so it's not like, we're extending GWAS. We're actually looking at, plasma protein proxies, genetically proxied plasma protein level. And so hence, we think we can discover some new variants, and we think we have to some degree. How well do these genetic variants actually predict variation of protein in plasma? Like, are they strong genetic instruments? And for some they are, like CD55, AZGP1, and a number of the novel proteins. They all, we have a pretty good handle on variation in plasma based on the genetic variants that we have. But for things like TREM2, where only 5% of variation predicted in plasma is predicted by, these cis-pQTLs, or ApoE, where less than 1% is predicted by pQTLs, they still come up as significant, probably because there's such strong, GWAS risk factors. But we don't have strong genetic instruments using this approach. So I know it's a little opaque, but hopefully you follow. So we have 12 of our 18 have been identified in Alzheimer's GWAS before. Listed here, these top ones, less evidence for, but there is some. However, again, 6 are novel. If we look at everything, 13 of our 18 are implicated in immune function, with a number of other processes implicated as well, cell adhesion, lipid metabolism, etc. We could split up these 18 proteins into networks using protein-protein interaction. And if we did do that, we see module 1, module 2, module 3 relevant to lipoprotein assembly, clearance, adaptive immunity and innate immunity, respectively. So we see, immune and lipids, so sort of the same processes that are enriched in 80 GWAS data here. And everything circled in red here is novel. How do these proteins in plasma, when we actually measure their levels rather than their genetic proxies, how do they relate to ADRD biomarkers in plasma? Well, we see a good amount of relationship here, focusing on the novel ones. CD55 seems to be strongly associated with p-tau181 and the rest of the gang of neurodegenerative and AD biomarkers. AZGP1, same thing, along with SCAR5. So, we can sort of get a sense, like, maybe CD55, because of its relationship with GFAP, and p-tau, is implicated in a specific stage of AD biology versus something else where we see stronger relationships like SERPA with A beta. And this is actually interesting because it's very, it's very specific to A beta. So we can try to get a sense of, okay, if they are operating on these processes, what processes are they operating through? And then we can go to cell-specific expression, and this is using databases that are available, through Tony West-Corey's lab, on a Shiny Tool app and Perch. It's super valuable, but this is single cell gene expression data. So we look up our protein, our genes of interest, and we find, and this is just focusing on some of the novel candidates, CD55 in Alzheimer's disease brains is, highly upregulated in microglia compared to control brains, many magnitudes higher. Same with little RB1. And when we look at little RB5, another novel gene protein that we discovered here, perivascular macrophage, not really upregulated anywhere except in AD and perivascular macrophage, and similar findings with ACGP1. So we can sort of get a sense of, okay, this is maybe how these peripheral proteins might be influencing CNS outcomes, or for some of these proteins, plasma abundance may be just a readout of what's happening in the CNS, right? And so we look at how these proteins in CNS, in CSF specifically, are related to this abundance of these same proteins in plasma. And we see for some, there's strong correlations. For some, there's almost zero correlation. And so, like I said, we could simply be getting, okay, this protein, little RB5, is relevant in the central nervous system because, it has a strong relationship with its level in the plasma, or just it's sort of the plasma level that we see is just an indicator and a biomarker of a process that's happening centrally. But we also have proteins that are expressed primarily peripherally, not associated with CNS measures of the same protein. And so we think that it's plausible that peripheral levels of variation in these proteins may actually be the drivers of disease itself. So I mentioned that we looked at African-American PQTLs in ERIC and applied them to an African-American GWAS. And so we had the opportunity to do more genetic discovery in African-American, using African-American GWAS data, and find independent variants that weren't identified in the original GWAS itself. So here's what we found in this volcano plot here. ApoE did come up as significant, as we might expect. And we found a number of others that didn't reach genome or proteome-wide significance in this case, but were significant, at least nominally. And among these is TREM2 and a number of others, which, I've heard BCHE and MMP12, but the others were at least new to me. And we can see their biology is mixed. It's not as specific to immune function as we saw with European American variants. But what was really interesting, in my opinion, was when we looked at how, and this takes some gymnastics, so bear with me, we looked at how the proteins are identified in European Americans, their genetic variants, how they predicted the Alzheimer's risk using the African American GWAS. We can get a sense of like, okay, are these things that we discovered or that we replicated in European Americans, do they apply to African Americans? And largely the answer was no. TREM2 came up, complement receptor 1. So these are well-known risk genes for AD. They seem to be relevant, at least using this approach to Alzheimer's disease in African Americans, but many of the others did not. As you can see here, sort of maintain associations along this horizontal. We did the same thing in the reverse direction. So we have these about 12 proteins, genes that we think are relevant to African Americans. Are they, when we apply their variants to European Americans or European people of European ancestry, are they relevant? And TREM2 is, but a lot of the others, and given their associations being along this vertical, don't seem to be relevant in European American GWASs. So, and this one we just didn't have PQTLs for. We know ApoE is relevant for European Americans. So, yeah, I mean, bottom line is we don't see a lot of genetic overlap between, at least genetically proxied protein level in terms of their association with Alzheimer's disease risk in European Americans versus Alzheimer's disease risk in African Americans. So, we have, all in all, we have at least 6 genes that we think are novel that are potentially operate through their influence on plasma protein abundance, or their level of plasma, the level in plasma may be a readout of what's happening in the CNS. Primarily, they're seem to be immune related, cellular adhesion related, and seem to be operating, if they are mechanistically relevant, through microglia and macrophage. So the next question is, okay, because we found these peripherally, we think this is potentially a peripherally mediated process. Can we modulate these proteins peripherally to actually modify disease? And so that's where we're left. We're taking some steps to try to test that specifically with collaboration, but we don't yet know the answer to that question. Okay, so I want to breeze through this because it's important. I want to get to it, but I also want to leave time for questions. So, I'm just going to go real fast. Here, what we did was we tried to identify biomarkers for blood-brain barrier dysfunction, brain endothelial cell breakdown specifically. We're familiar with the blood-brain barrier, so I'll breeze through this, but we were focused mainly on brain endothelial cells in this analysis and identifying proteins that seem to be specifically given off by brain endothelial cells, given their primary role in the BBB. And we then determined, okay, are these BEC, brain endothelial cell specific proteins, relevant pathologically and clinically? And so here's sort of the layout of what we did. This was a fully translational project that was led by Jennifer Cordon and Michael Duggan, published in Stroke last year. But first we used, again, going back to GTEx, going back to Human Protein Atlas, identified specific transcripts from our we're looking at 7,000 proteins specifically. And so we identified transcripts for those, examining which of those were specific to endothelial cells. And once we had 48 that seemed to be specific to endothelial cells based on a threshold, and sort of a calculation that was developed by Ananthi Kucha, featured here, we identified the 28 of these 48 that were specific for brain endothelial cells using available data from single-cell RNA-seq data, which can, through that, identify transcripts that seem to be unique to BECs. So we took these 28 that we think are genes that are specific to brain endothelial cells. We have their protein level. So we looked at, okay, are these the proteins that are coded for by these genes? Is their protein level associated with white matter lesion volume, which we think of as a pathological expression of BBB breakdown? And so we found 5 that met that, cut that criteria. We took these 5, created a composite score. We took through 3 out to improve generalizability because a lot of cohorts didn't have the 2 of the proteins we're looking at. But using these 3 proteins, we built a model that was able to predict about 11% of variation in white matter lesion volume in the BLSA, and you see a stepwise increase based on quartile. So we validated this externally. We took our proteins, our BEC specific proteins. We took our composite score. We asked, is it predictive of white matter lesions, microbleeds, and lacunar infarcts in the ARIC cohort? And we found for CDH5 and RSPO3 and our composite, that seemed to be the case, and RSPO3 extended to microhemorrhages beyond, extended beyond white matter lesions to microhemorrhages. But importantly, our composite score maintained its predictive value here for white matter lesions. And then we showed it was predictive of future dementia as well, suggesting there's some clinical relevance. So, just wrapping this up, we found 28 brain endothelial cell specific proteins, really using publicly available data with the total pool of potential proteins as those that were on the most recent proteomic platform. So because we could measure all of these proteins, we were able to relate them to clinical and pathologically relevant phenotypes. And we found that cadherin 5 and RSPONDIN 3 in particular validated in an external cohort, as did the composite score itself. Okay, and we know a little bit about the biology. CDH5 has actually been implicated in CSF. So I was excited to see that in plasma, this may also be a blood-brain barrier biomarker. So with that, I'll end. Thank you for listening. And hopefully I can answer some questions. Before I do, I just want to thank all the people who made this possible. Obviously, this is highly collaborative stuff and couldn't be possible without my lab, the ERIC individuals and participants, and the BLSA participants and funding, of course. So I'll stop there and I'm happy to address questions.
Wonderful. Thank you so much. This is so exciting. If anyone has questions or comments, please add them to the Q&A. We still have a couple of minutes. So yes, please do that. Meanwhile, I actually have a question. You started by talking about these risk factors, and so I'm wondering if you've been able to look at how hypertension and these other factors that are known to influence immune system and inflammation, how they are contributing to your findings.
Yeah. So we haven't looked at that directly. So these vascular risk factors, I mean, what we've done with these vascular risk factors, we know they influence the proteome, right? So we've controlled for them just as covariates because we want proteins that are independent. But we also have a separate paper that was published in Annals of Neurology last year that actually proxied like cardiovascular risk based on the proteome. So we could, we found a set of proteins that were predictive of a bunch of different health factors, cardiovascular risk, secondary cardiovascular risk, heart failure. And so we could compute quantitatively risk for each of these outcomes, even in people who didn't actually have the clinical phenotype. And so we thought that was very powerful. So then we applied these to, predict, we use these as predictors, and we found that even in people without any cardiovascular disease, if you had the proteome signature indicative of future cardiovascular disease, you were at risk for dementia risk, but also volume loss, A beta, p tau. Yeah. And yeah, it was a cool paper, very unique approach. But yeah, if you email me, I could send you the link to that. I think you find it interesting based on your question.
Oh, absolutely. I will send you that email. And have you noticed or have you looked into any sex differences?
Sex difference? We have, and the 2023 paper, the one that I talked about initially, when we find our candidate proteins, we typically look at them to see if there's stability or differences, effect modification by sex. And so we did report those findings. There wasn't a whole lot of difference by sex, at least in those top set of proteins. But I imagine, had we done stratified analysis from the start, we would identify proteins that were much more divergent by sex. But just by virtue of making it to the top, these proteins had to show similar directionality across the sexes. But it hasn't been a focus yet. But I think there's plenty of opportunity, to look at sex differences using some of these approaches.
Yeah, absolutely. That's very interesting. All right. Anyone has any questions or comments?
I don't know if you can see me. I have a question, or more like I'm thinking about like future directions. So a lot of the work that you have done is kind of like thinking of dementia risk and also like Alzheimer's disease. But I was wondering if in the future you are planning to do something that can speak more about the specificity in terms of the dementia, like thinking about Alzheimer's disease versus vascular dementia or frontotemporal dementia and other types of dementia. And if that is in the plans, like how will you actually go about doing that kind of research?
Yeah, it is in the plans, and we've just been limited by, well, the cohorts that we've been working with, which are fantastic, but because of their size, we haven't been able to do a deep dive on each individual on potential etiology. Most of the people are diagnosed with dementia, don't have scans and didn't have fluid biomarkers. Now that we can apply, use plasma biomarkers to sort of estimate amyloid status, we think we can really get a better sense of like, okay, for this person who developed dementia, is it likely, is Alzheimer's at least contributory? So that's kind of how we want to do it more broadly with this cohort. The alternative is just using cohorts that have etiologically defined dementia risk. UK Biobank, for example, they have a different proteomic platform. And, who's to say how accurate those etiologic diagnoses are? But when we do look at protein associations by etiology, we do see some a lot of variation. Our study is, shows a 4 times higher association with vascular dementia, than it does with Alzheimer's disease. So, we wouldn't know that unless we looked at it in UK Biobank. I didn't present those results, but we did look at it, and so now it's like, okay, maybe GDF15 doesn't really play a role in AD. Maybe it's a driver of cerebral small vessel disease or something underlying BAD. So it's incredibly important to look at etiology.
Thank you.
And actually, sort of following up on Jaquel's question, I thought the litter genetic overlap between the European and African American cohorts was really fascinating. Are you planning, or do you have access to other cohorts with other ethnic and racial backgrounds?
Yeah. Not in the immediate future. I don't, we have, I don't, let me think. We don't have it in the, like, we would love to apply these analysis to other cohorts. And they certainly could be applied as long as they have these large scale proteome platforms. We could take the PQTLs, we could take the proteins that we validated and look elsewhere, very easily. But it's really just about a sense of like what's available. And I encourage all cohorts to adapt like proteomics. It's been incredibly informative. And I know other cohorts are. Whether or not they're going to let me use the data, is another question. I see Steph has a question in the chat.
Yes, so she says, fantastic talk regarding etiology. Have you seen different modules, protein groups appearing based on age of onset that would give information on different mechanisms?
Based on age of onset. Yeah, so we haven't gone beyond, and hi Steph, I'm glad you made it. I hope you're doing well. We haven't seen differences beyond well, we haven't actually looked beyond what I mentioned, where the module, I forget, I think module 9 at this point seemed to be associated with dementia over a shorter follow-up time, versus others that were associated with dementia over a longer follow-up time. But I know it's not the exact same as looking at modules versus age of onset, but it would be interesting to look at like, module expression and time to dementia onset as an outcome versus, risk of dementia onset. And that is doable. So, thank you for that idea.
All right, well, it's already 1 o'clock. Again, thank you so much for this presentation. I look forward to seeing future papers coming out of your lab. And thank you everyone for coming and for your questions.
All right, thanks for having me. Take care.
Great weekend.